I try to imagine a fellow smarter than myself, and then I try to think, what would he do?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. I'm your host, Jesse Lawler, excited to bring you the 84th episode of this podcast dedicated to the improvement of your own brain by any and all means at your disposal. This episode is, I'm liking to think of it as kind of like the big summer movie action spectacular episode. It's that time of year, we've got all these big action movies coming out, and this is going to be probably as close as we ever get to doing an action episode of Smart Drug Smarts. We're going to be talking with a doctor whose job is specifically to research how to keep brains and cognition optimally functioning in extreme environments. When I say extreme, I'm talking about deep sea diving. I'm even talking a little bit about interplanetary exploration. We're going to be talking with Dr. Don Couragous of the Florida Institute for Human and Machine Cognition. If that name rings a bell, it's because the Florida Institute for Human and Machine Cognition is also the home of Dr. Ken Ford, who we had on several episodes ago. He's the founder. And if you didn't catch that episode, but it still rings a bell, it's because the Florida Institute for Human and Machine Cognition recently came in second place in the whole world in the DARPA Robotics Challenge. They had a robot called Running Man, which is the best robot in the US and apparently second best in the entire world. So a lot of interesting stuff going on over there, obviously, but we're going to be talking more about humans than robots in this episode, and that'll, of course, be in the main interview. If you hang around until the very end of the episode, I'm going to give some strong evidence that it's possible to have a memory that is too good. Kind of flies in the face of a lot of stuff we talk about. We think about how can I get a better memory, which compounds might improve my memory, but as we will hear, there is a sweet spot and it is possible to go beyond it. That'll be in the ruthless listener retention gimmick, but as usual, let's start off right now with This Week in Neuroscience.
Smart Drug Smarts, This Week in Neuroscience.
So I think you've heard me harp about it before, but one of my personal pet peeves in the world of neuroscience, or this is actually outside the world of neuroscience, is something in popular culture. I think that anybody in neuroscience knows that this is not true, but you hear it again and again that humans only use 10% of our brain, and if we could only use, blah, blah, blah. And I always feel like it should be patently obvious that we wouldn't have this galactically large brain so big compared to our body size versus any other animal out there if it wasn't being used for something. The brain is a very evolutionarily expensive organ. It forced us to have these big heads that were born premature compared to other animals. It's something like 3 pounds in weight, but takes up 20% of our body's total energy. There's basically a lot of brain tax that we pay for just having this thing. So if only 10% of it were getting used, that wouldn't make much sense. Hopefully this makes you as angry as it makes me, but scientists at MIT have just completed a study which helps give a little more firepower the next time somebody around you says, oh, we only use 10% of our brain, and you feel like giving them the smackdown. So a lot of this misconception comes from fMRI-based brain studies that show activation of brain regions based on the measurement of blood flow within the brain. Certain thoughts get thought or actions get performed, and there's a general net flow of blood from one area to another. Those areas sort of light up on the computer screen showing such things. And of course, the colors are carefully tabulated to show differences within the range you're looking at, just like you would adjust the contrast of a photo you're developing so there actually is contrast within the image. And then it looks like, oh, here's an area of heavy blood flow, here's an area of very light blood flow. But what these researchers are saying is that this gives a very incomplete image of what's really going on because there's always blood moving around everywhere in the brain, and really what you're just seeing is a net difference in those studies, not a total lack of blood flow to some areas of the brain. Now, a recent study just published in Science magazine took a different way of looking at brain activation. The researchers built an array of 108 electrodes to measure neural spikes across almost 3,000 regions in 6 different functional compartments within the brain. Research subjects were shown a group of colored dots moving either up or down, and then we're told to pay attention to either the color or the motion of these dots, and then to identify the color or the motion that they saw with a series of eye movements. And this won't surprise you, but basically there was activation all over the place within the brain, particularly the cortex, when these tasks were performed. The study's co-author Earl Miller, who's a professor at MIT's Department of Brain and Cognitive Science, explains, some areas may process motion more than color, some areas may process color more than motion, and sometimes you can see the information rising up in one area before the other. But generally, information is distributed all over the cortex. So there's just a little bit more ammunition for you the next time somebody flaps that dumb non-statistic about the 10% of the brain. Go ahead, shoot them down, and hold your head high. And in other news, we were lucky enough to pick up a 5-star review this week from listener Lickdo, who said on iTunes, "Great podcast says what?" And then answered his own question, "What? Hey, great podcast. I enjoy the information." Thank you, Licto, and thank you to everyone who has taken the time to rate us on iTunes or on Stitcher, or spread the word on any of the various social media platforms that are out there. That's absolutely the best thing that people can do for this podcast. Sometimes I'll say things like, tell a friend, but actually I should say, send a friend a link, because in this hyperlinked world of the internet that we all participate in, that's generally the most helpful thing for people. Remembering names, remembering anything, in fact, can be difficult, but if you've got a link right there in front of you, that makes it easy. So anyway, thank you to those of you who have helped seed the internet with some links to Smart Drug Smarts. That is hugely appreciated. If you haven't done so, I will remind you that we have a mailing list that will let you know about absolutely everything going on in the world of Smart Drug Smarts, the podcast, the website, and beyond. You can sign up for that at smartdrugsmarts.com or by texting the 3 letters SDS to the phone number 33733 if you are on a U.S. mobile phone. And of course, a big and continuous thank you to those of you who are on the list already. So I mentioned last week, and if you're on the mailing list, you already know this because I sent out an email on Thursday, but I said that we've got something big that we were about to announce, and right now, you are listening to it, this very moment is the ripping off the veil moment. So in the past 6 months, behind the scenes, we have created 2 nootropics stacks. Actual physical products, stuff that we're going to be selling for sale, a commercial venture, etc., etc. Now, if it seemed like we've done a better job recently getting episodes actually published on time, please consider that this is doubly impressive because the publication stuff has really just been the visible tip of the iceberg we've been moving, with a lot of stuff going on under the waterline as we get all this set up. So I think all of you who have been listening for a while now, you know that I love doing this podcast. I've been doing this for almost 3 years now. We've gotten more than a handful of reviews during that time saying things specifically along the lines of, you can tell that this is a labor of love. And that's definitely true. It's something that I've really, really enjoyed and that I want to be doing even more seriously, devoting even more of my time to. Now, with that in mind, I wanted to create something that would be like a daily super stack for myself, and that could quite honestly also be a Smart Drug Smarts flagship product, something that tied in with what we're doing and could become a financial water wheel to keep all of this going and expand it, allowing me to give it more time and more attention. Now, I'm fully aware that there are going to be some people that are going to groan and say, oh, Jesse's selling out. The podcast was better when there was nothing commercial about it, blah, blah, blah. And yes, I recognize that. I know that's sort of an unavoidable complaint, but I've just got to suck that up and deal with it because honestly, I think that opinion is entirely wrong in this case. I think that having a financial engine behind this podcast is going to make it a much better podcast. It's going to make everything that we're doing better because of the time, the resources, and the manpower that I'll be able to devote to it. And I want to explain this a little so everybody can sort of see and understand the big picture and know how serious I am about these 2 initial products. So there's basically 3 ways for a podcast to make money. You can either ask people to send you money, sort of the public television channel model. You can advertise a product or a service for somebody else, or you can advertise a product or a service that's your own. So I decided to go with the last one, and here's why. Those first 2 options, asking for donations or advertising for somebody else, you've got to earn 100% of your money from your own audience, period. There's no other way to do it, because if you're asking for donations, where are you going to ask except on your own show? And if somebody else is paying you to advertise their product, they're going to be using affiliate tracking codes or whatever to make sure that it's actually your audience buying their product, because otherwise, why are they paying you? So in either of those 2 situations, the podcast audience really is the only financial faucet that you have to draw from. So I wanted to make a product, and here's one of the main reasons. This company that we formed, Axon Labs, can have a life outside of this podcast. We're launching on this podcast, yes. Axon Labs only exists because of this podcast, also true. And of course, I'm going to be completely thrilled if a significant number of you decide to give us a try and become customers. But this is also a brand, the Axon Labs brand that we're going to be building, which is separate and apart from the Smart Drug Smarts podcast. And 6 months from now, I want a lot of people to be buying Axon Labs products that have never heard of Smart Drug Smarts and never will hear of Smart Drug Smarts. We've designed stacks to be useful for anybody with a brain, anybody with human physiology, whether or not they have any interest in listening to a podcast about neuroscience. So while I want Smart Drug Smarts to be a springboard for Axon Labs, I'd love you to try it, I'd love you to buy it, I'd love you to evangelize it. Ultimately, I think that the launch of this brand will allow me to continue to improve the podcast while actually doing less overt advertising on the podcast than any of the other models, because the Axon Labs brand will be selling to lots of people outside the Smart Drug Smarts audience. So I hope that made sense. I feel like that was a bit of a rant, but I wanted to kind of put that out there on the record so everybody understands where I'm coming from, and thank you for bearing with me. So what did we make? We made 2 amazing stacks. I'm not going to go into the full details on everything here because the ingredients and all that are available on the new website, axonlabs.io. That's not .com, that is .io. We'll be having an episode very likely next week about how we came up with the formulas, and that's not something that I want to just gloss over, but it's also not something that I want to spend a huge amount of time on before this episode's main interview. So here's the bullet points. We have 2 unique proprietary mixes. There is a cognitive stack called Nexus, which is built around aniracetam. As said before, aniracetam is my personal favorite of the racetams. It's been the chemical that I've been going to as sort of my go-to for creativity sessions. I find that aniracetam gives me clear thinking and a love of sort of the high-speed juxtaposition of different ideas. I've described it as a whiteboard drug. That's how I've always felt about aniracetam. There's also acetylcholine and other compounds in there as well. All of that's on the website. But one thing that isn't in there that I want to highlight real quickly is caffeine. I wanted to make a point that we're not putting caffeine into either of these stacks. A lot of companies that are doing premix stacks do that. I personally kind of feel like that's cheating. I've got nothing against caffeine at all. I drink it every day, but so does everybody. And caffeine has a stimulant effect that most people can feel. And I wanted to avoid that. I really wanted to ensure that when people feel our stacks working, that you know that there's something physiologically and cognitively distinct going on, and that you're not just getting something that you have available in every cup of coffee or every soda pop you drink. So what about our second stack? Our second stack is a mitochondrial amplifier called mitogen. Mitochondria, you may remember from biology class, are the organelles within cells that are responsible for creating ATP, basically the lowest common denominator energy source for cellular activities. Mitogen has both short-term and long-term effects. It provides chemical precursors to the mitochondria that you have now to allow them to do their job, and it also promotes the growth of all new mitochondria. So more energy now, more energy later, and this stack also has nootropic qualities itself because it's built around sulbuthiamine. If you remember a very early episode of Smart Drug Smarts, sulbuthiamine is a synthetic form of vitamin B1, which has been bonded with itself to more easily cross the blood-brain barrier as a dimer molecule. So I promise a lot more information about both these stacks in time to come. I'm going to try to gracefully straddle the line between promoting these things so you know how positively I feel about them, but also not being obnoxious about it and never trying to sound like an infomercial. So with that said, a couple of quick commercially oriented facts. I've got launch partners for Axon Labs who are going to be promoting both Nexus and Mitogen to the general public starting on July 14th. Until that time, we have two special offers that are basically just for Smart Drug Smarts people and my immediate family, because you guys are the only ones that know about this right now. First, any purchase made at axonlabs.io before the 14th will get free shipping to anywhere in the entire world. If it's someplace we ship to. We don't ship to all countries, but almost all of them. If you live in Kazakhstan, I might get screwed on shipping costs, but that's a chance I'm willing to take because I'm guessing that all of you hate shipping fees and almost none of you live in Kazakhstan. Secondly, during this pre-public launch phase, we have the super secret coupon code SmartDrugSmarts, all uppercase, all one word, and that'll knock $5.95 off the price of anything that you buy on that website. So both of these are modestly awesome little discounts, but please do not make any purchases just for the discount. Make the purchase if you do, because these are powerful, effective stacks. These are both things that, of course, I am taking myself, and knowing what a health nut I am, knowing what a cognitive enhancement fan that I am, hopefully that is enough to pique your curiosity. Okay, so that is that. Thank you for allowing me that time, and now let's move on to the main interview.
Smart Drug Smarts.
So as promised, we're about to be talking with Dr. Don Couragous from the Florida Institute for Human and Machine Cognition, which is an organization that I'm getting a little bit of a crush on based on the sorts of things that they're working on. They're doing everything from competitive robots to figuring out best practices for deep sea exploration, space exploration, all the kind of stuff that the nerdy little science kid in me gets super excited about. And honestly, it sounds like it's a sort of a jungle gym environment for nerdy little science kids that have grown up into PhD having science adults, and they're just working on stuff at the very highest level. So super exciting to get to speak with her. What Dawn is essentially tasked with is figuring out ways of making the human brain function as well as possible in the world's highest pressure environments. And that's high pressure in both senses, high stress and also physical pressure in under hundreds of feet of water in the case of the divers that she's working with. And she's a diver herself. So she's coming at this from a very hands-on first-person perspective and then using the tools of science to figure out how to best support people in these extreme environments. So if you will pardon the pun, let's dive in with Dr. Don Couragous.
When I was very young, I moved from Iowa at the age of 9, moved from Iowa to North Carolina, and went to the beach for the first time ever. I think I actually had gone to the California beach once before that, but I went to the North Carolina beach, and I was fascinated at that point in science, just in general. So I loved collecting rocks, and I was interested in geology, and I had a rock tumbler and all the books on rocks and fossils and dinosaurs. And so then I went to the beach and I was like, this is amazing, just seeing the span of it and just the entire ecosystem. I couldn't even wrap my head around it. And so I ended up at the age of 9, this tells you what a bit of a dork I am. I decided I wanted to go into marine biology. And so I asked my parents for Christmas, I wanted Jacques Cousteau's Ocean World. And so they bought that book for me for Christmas. That was my big Christmas present. And I carried that book with me in my backpack, on the bus, every day for a better part of a year. And I read every page, front to cover, several times. And so I was like, man, marine biology is the way I want to go. I want to become a scuba diver. So when I turned 15, I did my scuba training. I took a recreational diving class and just fell in love with it. I was just chomping at the bit to get underwater. About a year into my diving experience, I took a trip. So we were still in North Carolina at the time, and we took a dive shop trip to Florida, to the springs of North Florida, and just open water diving in the springs. And there's a place called Ginny Springs that has these beautiful underwater caves, and there's one in particular. It's a giant cavern that's graded off, and essentially it's open for open water divers, so people who aren't certified in cave diving. And my instructor took a couple of us in there. My poor father had to watch from the surface as his 16-year-old daughter, I think at the time, went underwater into a cave and disappeared into a hole in the ground, essentially. And I fell in love. I just absolutely fell in love. So I shifted from the ocean a bit. I was still doing a lot of wreck diving and ocean diving, but I shifted a lot to diving in the springs and caves around North Florida. And so I shifted my interest in research kind of from marine biology over to geology. I ended up changing my college major about 4 or 5 times, I think, from geology to physics to zoology. I found it all really interesting. And so I ended up going into biochemistry, which is kind of a nice brush over. I figured it could be applied across the board. And in the meantime, while I was in undergrad, I was working on cave diving projects. So at that point, I had conjured up enough experience, actually pretty extensive experience, cave diving, and started working with some of the best in cave diving. I sought them out mostly because I just wanted to learn from them and see what they were doing.
What is it about cave diving in particular that we're discovering that makes caves special, their own ecosystem, their own resource base? What is it that's the draw?
There are a number of different things. The first thing is just the very basic question of what's around the next corner, exploration. You know, for me, going down into that cavern the first time, and we swam down to the grate, and the water in the springs in North Florida, a lot of the springs and caves, is crystal clear, air clear. I mean, it looks like sitting in a room. And the white walls are kind of brownish white walls. The limestone caverns, just gorgeous, the way the light bounces off of them, your dive light shining around the cave. So that's one thing, just the beauty of the caves. All caves are different with respect to the height, the width, debris pile. You find a lot of fossils, a lot of interesting stuff. The life in the caves is really interesting. You have blind crayfish and catfish that have adapted specifically to the underwater cave environment and the darkness. So they're really interesting. You don't see a ton of them typically, but you see them around. But the biggest thing is just, like I said, what's around the next corner. And even if it's already mapped out and you're just going for a dive and something that someone's already explored, and you've even been diving in it 4 dozen times, it's still fascinating every time you go, because it's always kind of fun to peek around the next corner and just see what's in that environment. It's interesting to me because I see science as being very much along the same lines, what's around the next corner. And so that's kind of the continuing theme. That's how in my mind, I kind of tie it all together. So I ended up going in to cave diving. I worked for a nonprofit in North Florida for a couple of years. They do underwater dive training and also environmental science. And so worked with them while they got started. And that was a great experience. I got to travel a fair bit for my job and dive around the world. And I got the chance to see fascinating underwater wrecks, fascinating underwater caves. We did a lot of video sampling of different underwater wrecks, different underwater caves, mapping of those caves, sample collection for different research groups. And then the one group I worked with the most was a group exploring this deep underwater cave system we'll call the Springs. And it's just south of Tallahassee, Florida. The caves average just under 200 feet in depth. And our guys were doing exploration dives that were just pushing the limits every day. Even our support divers were pushing the limits compared to what a normal recreational or even a normal technical diver would be doing.
So for people that don't have diving experience, what are some of the threshold levels where a recreational diver wouldn't go beyond this depth and things like that?
Yeah, so typically the max depth is 130 feet for a recreational diver. And then there are various certification levels that can take you deeper. You train on specific gases, you train with specific equipment so that you can go deeper and be safer when you do that. Our guys were doing dives that were, for example, there was one dive that they did. It was average depth of just under 300 feet or just around 300 feet for 7 hours of bottom time. So that's just time at depth. The decompression profile coming out of that, I believe, was 21 hours. It took them about 21 hours to come up from depth, and that's so they can come up without risking decompression sickness because they've taken up so much inert gas in their tissues at that point. So that was actually a swim through. That was actually kind of my last hurrah with that project. I worked with them for over a decade, both as a diver and also as a dive manager. So they have been trying to connect two major cave systems for the better part of, I want to say, almost 2 decades at that point. And they finally did. And I was actually there when they made the connection. And so then they did a swim through. So essentially they went in one cave and came out the other. And they came out of Wakulla Springs. They went in at Turner Sink, which is down the road from Wakulla Springs. And like I said, it was about 7 hours bottom time. And it's just very intense, but very exciting and rewarding to see all that hard work pay off.
So 7 hours of bottom time, but then 21 hours to decompress. What is actually the physical environment of somebody in that decompression process?
They start their decompression deep, and as they work their way up, they have different gases that have been staged for them. So as they come up, the gases have more oxygen in them to help get rid of the inert gas that has built up in their tissues at depth. And so they breathe these different gases as they're coming up, and we have support divers that are around them at all times to make sure everything is going okay. Then when they get to some of the shallower depths and the oxygen content is higher or at its highest, including 100% oxygen, then they're in these overturned troughs that are put up against the ceiling of the cavern and essentially allows them to finally get out and either physically completely get out of the water, or at least stick their head up in a dry space. But for the last very long portion of the decompression, they're actually completely out of the water except for their feet kind of dangling in the water. And so they sit in there and they breathe the oxygen and they breathe another gas that kind of helps them relieve the stress of breathing 100% oxygen for an extended period of time, because that can actually be bad for your lungs over a period of time. So they switch back and forth on a schedule. And we bring food down to them. We brought, red velvet cake and hot food and all kinds of interesting stuff to them in tubes just to keep the spirits high. And so then they can also talk to each other, and they can talk to the support divers who stick their head up, although you don't want to stick your head up in there for too long because there's a lot of CO2 built up in there.
People have been doing diving for a long time, but the technology has been radically improving in the last couple of decades. Where are things at with sort of the protection of people's brains and the performance of people's brains as they're doing these dives?
When we look at the military diving population and the recreational diving population, it's relatively safe. The risk of accident, especially when you're looking at decompression sickness, particularly in the recreational diving community, is fairly low. But as we kind of push our limits and as we extend, so we have equipment that is more advanced, equipment that's allowing us to extend our bottom times, equipment that's allowing us to kind of push our limits a little bit, we're starting to get into a place where we want to make sure that we're also protecting ourselves on a physiological level. So right now things are relatively safe. The biggest risk is oxygen toxicity. So breathing too much oxygen is actually very dangerous for divers. So you can breathe like 100% oxygen for, say, an extended period of time, even clinically. It doesn't have to be divers. This could even be a clinical population. And that's not great for the lungs over time. It can actually lead to what's called pulmonary oxygen toxicity. And it's a scratchiness, and it causes respiratory difficulty. But with divers, one of the things that they also have to consider, and also people in hyperbaric chambers, although they try to mitigate the risk by minimizing how long and how deep they put patients on oxygen and hyperbaric oxygen exposures for clinical treatment. But for divers, one of the big concerns is if you breathe high pressure oxygen, particularly deeper than about 30 feet, you start leading to a risk of CNS oxygen toxicity. So this manifests as a variety of symptoms that can lead up to seizures underwater, which is obviously very, very dangerous for divers. So one of the big areas of focus right now, specifically in the military diving population, because they tend to use oxygen rebreathers. Essentially, oxygen rebreathers will scrub out the CO2 and kind of recycle the oxygen, so it extends the gas supply and also keeps the diver from, you're breathing 100% oxygen to minimize any kind of risk of decompression sickness, or essentially get rid of any risk of decompression sickness because you don't have inert gas uptake. So the problem is that they're breathing this high pressure oxygen at a certain depth, and they can have a risk of developing seizures underwater. So we're trying to find ways to protect military divers against development of oxygen seizures. Also, in the setting of submarine escape scenario, where they breathe 100% oxygen prior to the escape portion of the ascent to try to minimize risk of decompression sickness again, because they're breathing that oxygen at such a high pressure just before the ascent, there is that risk of seizure development.
Escape from a submarine. It sounds like a terrible case scenario if that would actually need to be executed. Is this sort of a fire drill that would happen, once every 20 or 30 years, or is this something that happens with any regularity?
No, it doesn't happen with regularity, thankfully, but they do train for this. So even when they're doing the training scenarios, it's ideal to find a way to protect the operators in that environment.
Something that I've always heard, and I wonder what the basis for this is that different people have a whole lot of variability as to how they perform underwater, how likely they might be to get something like the bends on an ascent. Do we know anything about why there is so much difference person to person? It seems like more so than in a lot of other physiological traits that we have.
That's a great question. There are a lot of factors that filter in. So between individuals, things like gender and age, there's still studies that are going on, and it's funny because the results tend to kind of bounce back and forth with respect to how important gender and also age are. Body composition seems to be important with respect to susceptibility. Individuals can have what's called a patent foramen ovale. So essentially this is an opening that's in the heart. You have it when you're in your mother's womb, and essentially allows the blood flow to bypass the lungs when you're in the womb. When you're born, and about 75 to 80% of the population, that closes completely. And about 20 to 25% of the population, that remains open to some extent. The problem with that is if you're diving, so a lot of divers will have bubbles that form, and you can actually see that. We've done a lot of field studies where we follow divers in the field and then done Doppler ultrasound or transthoracic echocardiogram to either listen for or look for bubbles that are passing through the vascular system. And most of the time, they're just a few bubbles, there aren't a lot, and they'll pass through the heart and be filtered out by the lungs. Not a big deal. You never see them, they never bother you. But for some people, there's a risk that those bubbles can pass through that opening and shunt over to the arterial circulation. With that, you have some serious consequences, obviously, if you have bubbles that pass over into arterial circulation and potentially get to the brain or the spinal cord.
What would be amazing is to see if there's been similar studies on diving mammals, seals, whales, things like that, if there's any commonality, because they might be getting these bubbles also.
Yeah, so they've done studies, and so what's interesting is the inert gas uptake is different. There are also physiological differences in marine diving mammals, and not only with respect to inert gas uptake, but also how their blood flows. But also, one of the interesting things is marine mammals do not have certain coagulation factors that we think might be implicated in decompression sickness and decompression stress. So it's called contact factor. Essentially, it's a coagulation factor that only kicks off if the blood has exposure to charged surfaces. So the idea, and this is proposed decades ago, that you have these bubbles and they're passing through the blood, and you have plasma proteins that are getting kind of caught up at the surface of the bubble and are becoming unfolded. So you have hydrophobic portions that are inside the bubble, hydrophilic that are outside of the bubble. And so you have this kind of charged surface running through your blood system, and that some people might have this exacerbated response to those charged particles that are passing through your blood system. Well, marine diving mammals do not have that contact factor. So there's probably a number of reasons that they're protected against decompression sickness. Actually, there are a number of researchers, including our group. We're working with several other collaborators who are looking at marine diving mammals and some outcomes from stranded whales and what they're finding in those animals, and then trying to compare them to human exposures.
You just said our group right there, and I don't think I've let you jump ahead to the current update to the story of where you're at right now and what you're doing. So maybe we should fast forward to the present.
I went from all this underwater exploration and research and working with great people along the way, and I realized we were doing these dives and we're also working on studies. So I would even participate, and a lot of our people would participate on diving physiology studies, trying to optimize the decompression protocols out there, trying to protect divers against like CNS oxygen toxicity, some of these other things that we've talked about. And so one of the things that was frustrating is there's still so much that was unknown. And so I decided that I was going to just kind of delve into the literature. I just started kind of peeking around. At that point, I was doing some marine biology research, and I had done a little bit of human physiology research at Duke University as an intern, but not too much. And I came up with an idea that I wanted to look at decompression stress in divers and see essentially what was changing in the divers. I wanted to actually look for an objective biomarker of decompression stress, which is kind of a holy grail for us. So I started looking for an objective marker, and so I essentially went in and what was at the time relatively new technology used gene expression microarray. And so essentially looking at all the genes in a sample of blood. So we extracted peripheral blood cells, extracted RNA from those, and then looked at expression of all the genes in the sample. So we did pre and post dive, and we worked with military diving exposures that were being conducted on an experimental basis. And we also controlled for high pressure oxygen breathing, time of day, and also exercise. So that was kind of my first project. I actually turned that into my thesis project, and that became part of my PhD thesis. So, trickle down the line, last year I met Ken Ford, who's the director of IHMC, and over the course of the year I came down and started giving some presentations and working with some of their researchers through a couple of meetings. And he eventually invited me to join IHMC to essentially create an undersea research program and also to kind of work from the side of optimizing human performance and resilience in extreme environments. So not just undersea operation, but also high altitude and a number of other extreme environmental exposures.
What are some of the commonalities there? Because it seems like at first blush, those would be almost completely opposite scenarios, high altitude versus undersea.
So what's interesting is actually when you talk about high altitude aviation, you have a quick reduction in pressure, especially certain cockpits aren't pressurized like to the same degree that, say, a commercial airliner would be. And so we've seen over time that some of these high altitude pilots are actually developing or have developed decompression sickness, just like divers. Just like divers experience a reduction in pressure, so they're taking up inert gas at depth and then coming up. And if they get to a place where that pressure in the blood and tissues exceeds that surrounding them, then they can reach a point of supersaturation and develop bubbles, essentially. So just think of opening up a bottle of Coke too fast and not allowing it to equilibrate well enough. So essentially the same thing can happen with high altitude pilots if they go up too fast or if they experience such a drastic reduction in pressure.
What do you find as far as functional cognition in these extreme environments? When we do things right, are we able to keep people performing essentially as we would expect cognitively?
Yeah, I think so. I mean, you're talking about a lot of stressors that are taking place, so I don't know how much of it is at the cellular or molecular level. You're taking in a lot. So, it's kind of you almost reach a bottleneck, essentially, of cognitive input. Say for a deep dive, you go in, you've got all of the different environmental considerations that are impacting you and just how you feel in general. So you have temperature, you have immersion, you're breathing a denser gas, a number of those different things. Plus you're having to multitask upon multitask. So you're reading multiple gauges, you're handling multiple gases, you're navigating, you're following, making sure that your teammates are okay, and you're checking all those same things with them. It's almost like you get to a place where I think if you're trained well enough, a lot of that stuff becomes muscle memory. And so it doesn't reach a bottleneck to a certain degree. But I do think you're really pushing the limits with respect to what you can actually process on a continuous basis. You think about those guys I was talking about doing the 7-hour dive, that's a long time to be on, like full on. And so once they get to decompression, they have other people that are helping them out. But I think at the pathophysiological level, at least if you breathe in the right gases and everything else, you're not having any kind of major issues, at least.
You've started to do a little bit of research into what's actually involved for astronauts as they might be going on a moon mission, a Mars mission, something like that. What do we know about the effects on cognition as humans travel into space? And the thing that makes me curious is the pros and cons of why we should be using humans for these things when we can design robots that are structurally perfect, at least as best as we know right now, for the environments that we're putting them into.
So I think that as far as why humans versus robots, it's funny, I work at IHMC, which is very well known for their incredible robotics, and I don't know anything when it comes to robotics engineering. And I'm saying this from the human perspective, so it's kind of funny because I think Ken brought me to IHMC because I am coming from the human perspective. And as a human explorer, I'll tell you that at the end of the day, there's still the romantic notion of having humans go where humans have never been before. And I think that when you're talking about robotics, there's still a fair amount of human interaction that's necessary with robotics. So I think that robots will still be very much implemented in these long-range missions that we're discussing. But I think it's going to have to be a tag team approach between the humans and the robots. And so a lot of the work is going to be optimizing that human-machine interaction. So what's interesting is we also have potential for decompression sickness in astronauts. And that's during extravehicular activity, also known as spacewalks. When they go into their suits, they experience a reduction in pressure from where they are into the suit. And essentially they can develop decompression sickness once they transfer into the suit. Plus they're going through a lot of really strenuous motions during their tasks on the EVAs. Several of my colleagues back at Duke University were doing research in this area, and also Mike Gernhardt actually heads up that research at NASA. He's head of the EVA program there and works extensively with several different institutions, including Duke, to optimize EVA protocols. So essentially, right now they have it where the astronauts go through a series of exercises and also breathe 100% oxygen, which actually high-altitude pilots do as well prior to a high-altitude flight. And that kind of gets rid of that inert gas that I was talking about that's in the tissues, and also just kind of gets everything moving to try to minimize the risk of decompression sickness. So that's one thing. And then another concern is once we go into some of these long-range missions and get out of lower Earth orbit, what kind of radiation exposure are the astronauts going to experience, and what kind of impact is that going to have with respect to brain health and cognition overall? So these are all areas that are being explored by our group and by a number of different groups, and just trying to make sure that we minimize that risk and optimize cognitive performance on those long-range missions.
Question on that. The amount of electromagnetic shielding that Earth gives, when astronauts have been traveling back and forth to the moon in the past, how similar is the level of exposure that they're getting there versus sort of being in the empty space between, say, Earth and Mars?
That's a really big area. So not only trying to figure out how much and trying to extrapolate and trying to develop models on the level of exposure that they're experiencing, but also it's a very different type of radiation compared to, say, clinical radiation exposure. So the injury might be similar, but it also might be very different. So we're still trying to figure all of that. I say collectively as a group of researchers in extreme environments are still trying to figure that out.
You know, one of the things I always find humans stubbing our collective toes on is we tend to only really learn well when something goes horribly wrong, and then we're like, oh, in case that happens again, we should set up all these procedures. Have there been any cognitive disasters in these physically difficult environments that have led us to some of these revelations of what we should do now, how we should prepare?
Very interesting that you asked that. It's funny, I was actually just unpacking my boxes since I just moved here recently. So I was unpacking my boxes and going through my books, and it was funny, I found one of the books that was essentially the Bible for cave diving when I first got into cave diving, and it's called Blueprint for Survival. It was written by one of the godfathers of cave diving. His name is Jack Exley. And essentially he did exactly what you're talking about. He took a step back. This was, I don't know when it was written. I want to say in the 80s. It may have been even earlier than that. Very, very experienced diver, one of the best that was out there, incredible teacher, incredible explorer. And so he took a look. He said, okay, I've had these, especially back then, a lot of people were dying in underwater caves, as you can imagine. So there wasn't a formal training process. There wasn't, and typically trained divers fare very well. It's the people who are untrained that go into an environment like an underwater cave that quickly get into trouble. And so he started looking at particularly not just people who are untrained, but also trained divers, and saying, what happened? They died, unfortunately, but what can we take from this so that we can learn and pass that on to future generations? And so a lot of those were based on multitasking, but also a lot of it was complacency, getting a little too comfortable with certain procedures. And you can see the same thing with airline pilots. A very good friend of mine, his name is Gareth Locke, he's over in the UK, and he was working with the British military, and now he's working actually on his PhD thesis, but he's developed a human factors program specific to diving. And essentially he's doing exactly what you're talking about. He's going through accident analysis and saying, what can we learn about these accidents? What can we learn about what happened with respect to decision-making processes and cognitive performance? And how can we take that and apply it for future training? And he talks about the Swiss cheese effect. Essentially, one thing after another kind of falls into a place, and where all the holes line up, and that's where you have a disaster. And they've seen the same thing with pilots, and even they've seen the same thing in the medical setting, is that it comes down to complacency. You get a little too comfortable, and then something happens. You're having an off day, you've got some emergency comes up, something kind of throws you off just enough where you make that first mistake, and then it just spirals from there. People I know who do research in those different fields, they're all talking essentially the same language. So I think, it comes down to cognitive processing and, just kind of keeping things together to the right point where things are done either by checklist or some kind of task by task basis to make sure that everything gets done, that complacency or something doesn't kick in.
Airline pilots kind of famously have the checklist that they need to go down before they take off. And they've almost sort of removed the human from that process, or at least like human creative thought from that process as much as possible, and really made it just sort of a rote, you go down the list and you do these things. Where do you find the balance on something like a checklist-based methodology versus people using their own creative impulses and intuitions?
It's a great question, too. I think essentially the checklist comes down to, anything that is kind of standard operations. So for divers, say we have a rebreather. I was talking about the rebreather technology before, which is much more advanced, comparatively speaking, to a basic tank and open water scuba setup. There are a lot of moving parts and pieces. There are different gases. You have your scrubber set up. You have a lot of them have computers that are associated with them. And so there's been a big push for checklists and for divers sticking to their checklists when they're setting up their rig. And a lot of times we find that rebreather accidents, the same thing will occur when somebody kind of jumps off of that wagon and just starts, like you said, creative thought versus, sticking to the standardized approach. So I think with something like that, or just like a pilot, I don't want a pilot being creative when he's going through a checklist and getting ready to take off. So, we want them sticking to a standardized procedure. But I think then you get into a realm of different areas of exploration, or even when you get to a point of like maybe a situation comes up where you do have to have a little bit more creative thought about how to work your way around. That's where experience kicks in.
Let's say that somebody is underwater doing an exploration dive, and for whatever reason, something does go technically wrong and they find their cognition fading. Something about their own perceptual reality starts to feel like, wow, my brain is not functioning as well as it once was. What are the protocols for that? If you kind of realize like, my brain is starting to sputter.
So this is where I know people that do solo diving, even in extreme environments, which is something like what you just explained is exactly why you want to have at least one buddy, if not two, with you. This is why you have a co-pilot. This is why, you always have somebody else there so they can help you. Essentially, the protocol would be to flash down, to signal your partner, your dive buddy, and say something's not right. And we have a series of hand signals for everything. We even have, for some of the more complicated dives, we'll use underwater notepads to write things out if we need to, if it's more complicated than a hand signal. But I will tell you that most divers, if they've been diving together long enough, they can tell. But essentially you tell your buddy and then you figure out how to handle it. And a lot of times if they're clear-headed, then you let them handle it instead of trying to make things worse by handling it yourself.
Smart Drug Smarts.
So thank you so very, very much to Dr. Don Couragous for taking the time for that conversation. It really is interesting to me that here in 2015, with so much technology, so much research, and so much history with things like deep sea diving, that we still are kind of figuring things out that one might think we would have studied a while ago. But of course, better late than never, and a good time to be doing it now that we're getting such much better ability than we've had in the past to look at what's going on within brains in real time and be hyper analytical about what we actually find. So while not many of us will actually ever be in a sinking submarine, God forbid, I think the spillover effects for general society on the research that people like Dr. Kernagis are doing could be pretty profound, understanding how to best inoculate our brains against harsh environments and unforeseen traumas and neurotoxins and things of that nature.
Smart Drug Smarts. Ruthless listener retention gimmick.
So normally it's a pretty big source of pride to have a good memory, and those of us who are interested in cognitive optimization are interested in improving our memory. Figuring out tricks like mnemonic devices is cool. Finding compounds like Bacopa moneri that seem to physiologically improve our ability to capture memories is cool. And of course, remembering where you put your keys is also cool. But as we with so many things, there seems to be too much of a good thing, and there's a fairly recently recognized syndrome. Like literally the past 10 years is when science sort of accepted this as a thing. It's called hyperthymesia, and a hyperthymesic is somebody who accidentally automatically records pretty much everything into their long-term memory. Hyperthymesia literally means excessive remembering. These individuals can recall almost every detail of their lives. They're oftentimes excessively fascinated with dates. They can tell you what date anything happened on. But unlike autistics and things like that might calculate a date backwards with incredible accuracy, they actually just remember it. And this all happens unwittingly, automatically, like a memory savant. This term was just coined in 2006 after an article in the journal Neurocase. And that pretty much put it on the map as a type of brain disorder. I'm not sure that disorder is actually the right term, but I guess so. And since then, a lot of people have come forward and said, yeah, my brain is like that too. Apparently, not all these people check out, but a decent number of them do. There's several dozen people known worldwide to have hyperthymesia at this point. Interestingly, it seems like it's generally about 10 years of age that this automatic recording of memory switches on. It doesn't start from the moment of birth, and it doesn't even really start from the moment of a person's first memory. Most of us have our first memories at ages 3, 4, but the full vacuum cleaner memory mode seems to go on for people with hyperthymesia at about age 10. Hyperthymesics studied at the UCI Center for the Neurobiology of Learning and Memory were found to have brains structurally different from those of control subjects, including more robust white matter linking the middle and front parts of the brain. Surprisingly, these people did not score higher on routine lab memory tests or when asked to use rote memory aids. But when it came to public or private events that occurred after about the age of 10, says Dr. James McGaugh, they were remarkably better at recalling the details of their lives. The study also found statistically significant evidence of obsessive-compulsive tendencies in these people. Many of the individuals sampled have large, minutely cataloged collections of things like magazines, videos, shoes, stamps, or postcards. And as much as most of us kind of fantasize about having a better memory, the people with this condition tend not to be such big fans of it. They spend a lot of their time kind of lost in memory because almost everything reminds them of something else or multiple something else's. And of course, memories of bad things don't necessarily go away either. So the inability to forget, and we talked about this on a past episode, how the inability to forget can also be a major problem with post-traumatic stress disorder. That obviously would be compounded for somebody with hyperthymesia. Last but not least, it is worth mentioning, there's also a pretty good, apparently, British documentary on Channel 4, that's Britain's version of PBS, called The Boy Who Can't Forget, and that details the life of a British-born university student named Aurelian, who is a hyperthymesic. So if you like your neuroscience documentaries and you have access to Channel 4, check that out.
Smart Drug Smarts, the podcast so smart, we have smart in our title, twice.
Okay, that is the end of episode number 84, our action movie special edition summer blockbuster episode of Smart Drug Smarts. Thank you for hanging out until the credits are rolling. As usual, if you liked what you heard, please do recommend this podcast to your friends. And of course, check out smartdrugsmarts.com, where we will have the show notes for this episode and everything that we talked about. One last reminder that we've got a brand new website up at axonlabs.io. It's not .com, it's .io, and that is the home of our first ever, very own, honest-to-goodness Smart Drug Smarts products, Nexus and Mitogen. I will be back at you next week, same time, same podcast, and with the same unflagging commitment to helping you fine-tune the performance of your own brain. Have a great week, and stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.